Welcome, everyone. Thank you for joining us for today's webinar, The Missing Link to Improving Efficiency and Productivity in the Digital Lab, presented by Jay Campbell, Chief Commercial Officer, Somark Innovations. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Jay Campbell. I will now turn the presentation over to him.
The chain of custody in our labs is the unbroken link between all the objects used in your research, your people, the animal models, cell lines, reagents, compounds, the lab instruments and devices, the data records where data is stored, the link between the analytics and the data collected. In the SmartLab, there's no place for uncertainty. SMART requires your research and data to be specific, measurable, auditable, repeatable, but preclinical research is a long way from being smart. To give you two examples, in preclinical research, the use of rodents accounts for over 70% of all in vivo research, and the most common methods of animal identification are permanent pen marks, ear notches, and metal ear tags. Research has shown these methods of identification are prone to misreading errors at rates between 20 and 40%. In addition to animal models, the NIH recently reported significant levels of misidentification with cell lines. The lack of reliable identification of inputs to your research is further compounded by the lack of data quality control arising from the use of paper lab notebooks and spreadsheets, manual recording of experimental data, manual input of results data. They leave the process open to human error, transcription issues, and do not provide audit trails and lack transparency. So the lack of confidence in your data quality arising from your analog methods of identification, poor data management, and limited rigor applied to the process of experimental design, execution, and analysis is really creating a house of cards. The wrong mouse, the wrong weight, the wrong tumor measurement, all leading to the wrong conclusions, which is certainly evident in the in vivo preclinical research. The missing links are at the heart of the reproducibility crisis we see in vivo research. So how do we ensure SmartLab is Alcoa compliant? And don't forget CCEA too, complete, consistent, enduring, and available. At Somark, we have worked with over 300 customers over the past 6 years to better understand what makes a lab smart and how to deliver that capability in an affordable way. We believe the answer, or the missing link in the lab that prevents us from being smart, efficient, accurate, transparent, rigorous, reproducible, is digital identification. Digital identification is the technology that is connected seamlessly to automated data collection, processing, and analytical processes. At the heart of the Somark SmartLab is a common method of digital identification. We propose the adoption of the RAIN RFID standard for identifying all your lab objects, including your staff, your animals, your samples, and your devices. Passive ultra-high frequency tags are already used in billions of devices and extensively in logistics to track assets in real time and space. It's a proven low-cost, accessible, and scalable technology platform already at the heart of Industry 4.0 and will become part of the standard in the Lab 4.0 revolution too. At Somark, we have developed the world's first ultra-high frequency animal tag. This provides preclinical in vivo research, a digital method of identification that is low cost, more humane for the animal, and most importantly, is interoperable with other brain RFID tags that can be used with cage cards, samples, and lab instrument assets tracking. So you may be using current available RFID technology in your labs for animal identification, but these are not the same ultra-high frequency or RAIN compliant. For example, they would not integrate with a sample or a personnel identification badges. Additionally, the current RFID tags are not purposely built or designed specifically for animals, which is an animal welfare concern. So now you have solved the missing link in your lab, and all your objects are digital. You have to be able to connect those objects to your research processes, the right people, the right experiments, the right data records. To solve that challenge, we developed SensaLab, which is a technology platform providing tag connectivity to lab devices and animals and other inputs to a cloud-based data system that correctly stores the records and maintains full audit trails. SensaLab is built to deliver automated processes across the entire chain of custody. All you need is to tag your objects, plug in your readers and devices, and connect to the internet. SensaLab enables you to move away from paper and Excel sheets to secure, scalable cloud environment, and will connect with the technology you already have, your existing devices and systems, if you're lucky enough to have good data systems. So what does this look like in practice? Utilizing our low-cost benchtop tag reader, or you could buy a handheld reader, our device enables you to turn your existing lab instruments into Internet of Things devices by connecting them to our iConnect system. The iConnect captures the ID of the person, the animal, the object, and the data from the lab device, and automatically puts the data in the right record with an accurate time and date stamp in the cloud database. One of the key steps towards implementing the SmartLab after adding digital IDs to your objects is to use systems like iConnect to then automate the collection of data from experiments and post them to the right records in the cloud database with a full audit trail. That's what iConnect does. In our SmartLab, we don't simply upload a spreadsheet into an electronic lab notebook. We use SensaLab to ensure every data record is tracked and versioned independently with a full audit trail, recording where it came from, who recorded it, when and from which digitally identified subject, for example, a mouse, or what other inputs are associated, such as which cell line or compound. Remember, simply uploading files to an electronic lab notebook does not give you full control over the source data in the files. And one of the benefits of moving to a SmartLab is not just immutable data. It's actually much more efficient. We recently conducted 12 pilot studies across US and Europe. Results were 69 to 74% reduction in time to take data, with a 0% read or data input errors. Not only did we achieve process efficiency, now you have a smart lab with efficiently managed digital data. And now you may ask, so what? Well, the real value comes from the analysis of the data. In the late 80s or early 90s, there was an often quoted saying in the early days of data science, and I'm sure you will recognize it, and that was garbage in equals garbage out. There is significant excitement, and rightfully so, about machine learning and AI. But if you're training algorithms on poor data quality, really, how good are your algorithms? I have provided 2 reference articles in the presentation that focus specifically on this crisis in science. The key strategic advantage of a smart lab based on digital ID and automated data collection is the data authenticity, accuracy, and reliability. And what you will see is the higher quality of data will drive insights faster. You can even start to move towards near real-time discovery of exceptions, of errors, of trends. IDBS, a laboratory information technology company, estimated that saving 1 week in drug discovery process is worth $6 million. One of the advantages of this move to digital data is you move away from using Excel and manual manipulation of data preparation and transfer. The minute you download data from your repository, again, you open up a chink in the chain of custody. So the final missing link that stops the chain of custody from being broken is to keep your research data in a secure data environment and actually to point your analysis tools at it, preserving the integrity of the data. We propose using data visualization tools. You can gain insights into your data quicker. Tools such as Power BI or Tableau enable you to set alerts when they detect anomalies in data, preventing your research going out of control, losing animals to ill health unnecessarily, and will save you time and sometimes the value of your research. If your data is locked in paper notebooks or files on servers or even files uploaded to an electronic lab notebook, you will struggle to develop reliable analytical insights. And of course, when you start to apply predictive analytic tools, machine learning, and AI to augment the scope of your analysis and identify hidden correlations you did not expect, basing these discoveries on reliable, accurate data with your digital IDs at the heart of your data integrity will begin to minimize false positives and false negatives and ensure reproducible research. In conclusion, the SmartLab requires a range of capabilities to be in place. We have talked about the need for secure, scalable data management platforms, automated data collection devices, and data visualization and analytical tools. But at the heart of the SmartLab must be the digital ID. It needs to be consistent, integrated identification method, in our view, based on the RAIN RFID standard, passive UHF, EPC Gen 2, ISO 18000-63. Without it, the link between your research and data quality is at risk, and your chain of custody can break all too easy. Thank you for your time.
Thank you, Jay, for that informative presentation. And I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.